Alright, so now let's take a look at the next section, and this is on the paper methods, and it's going to be on, the first part of it is going to be on the isolating the, quote, virus. So now let's take a look at the next section in the above paper, which is on allegedly isolating the, quote, virus, and this paper starts off over here. So, isolation of virus, bronchoalveolar lavage fluid samples were collected in sterile cups to which virus transport medium was added. All right, and remember that this bronchoalveolar lavage fluid is they just put a tube down the throat and put a fluid inside, and then extract that fluid and all the material inside in the lower respiratory tract. All right, so they put this in sterile cups to which virus transport medium was added. And so virus or viral transport medium, VTM, is a chemical preservative used to preserve, quote, virus specimens during transport instead of using cumbersome supercooling equipment. And typical VTM components may include saline solution, just a salt solution, phosphate buffered saline PBS, or fetal bovine serum FBS. Yes, that's interesting. A buffer solution is an acid or base aqueous solution used to maintain a constant pH when a small amount of strong acid or base is added to it. And acids and bases are defined as in the three theories shown in the image below. So here's classification of acids. There's three different theories of them, and which some of the theories overlap with the other ones here. So classification of acids and bases. So acids, you'll have a Lewis acid accepts electron pair. Then Bronsted-Lowry acid donates proton or H+, and then Arrhenius acid dissociates into H+ when dissolved in water. And then classification of bases, you have the opposite Lewis base donates electron instead of accept. This one's accept, this one's donate. Then Bronsted-Lowry base accepts this proton. So this one is, accepts it, whereas this one donates this electron pair over to this. And then the Arrhenius base dissociates into OH- when dissolved in water. So this goes to OH. The other one goes plus. So acids go into plus in water. This one OH- dissociates into that. All right. Let's go back up here. All right, and now the pH or potential or power of hydrogen indicates the acidity or basicity of an aqueous solution. The higher the value, the more basic, and the lower, the more acidic. And an aqueous solution is a solution in which a solvent is water. And a solvent is a substance that dissolves another substance called a solute or solute, and resulting in a solution. And then this, lastly, this fetal bovine serum, FBS, is extracted from blood. Is extracted from blood drawn from a bovine or cow, bull, or oxen fetus after it is killed at a slaughterhouse. That's pretty wild. And then serum is the part of the blood not involved in blood clotting. So they would extract that serum from this fetal bovine, or this fetus from this cow, bull, or oxen that's killed at a slaughterhouse, and then mixed in with this, and may oftentimes be used as this viral transport medium. So then they would put these samples inside this serum from this slaughtered fetus. All right, so that's quite baffling. Continuing further in the paper, samples were then centrifuged to remove cellular debris, and the supernatant was inoculated on human airway epithelial cells, which had been obtained from airway specimens resected from patients undergoing surgery for lung cancer, and were confirmed to be special pathogen-free by NGS. All right, a lot of steps in this quote virus isolation. So centrifugation involves rapid spinning to separate objects by density, and the supernatant is the remaining liquid after centrifugation or settling. Inoculation is the introducing of microbes into the sample. In other words, you're, quote, infecting this sample. Resection is the removal of all or a key part of an internal organ or body part. Again, this is terms for surgery in general. Cancer refers to abnormal cell growth with the potential to, quote, invade or, quote, spread in the body. And specific pathogen-free, SPF, refers to being free of specific pathogens. And lastly, this NGS, that actually let's put this in bracket right here, next generation sequencing. Yeah, so in other words, they just did a genetic sequencing, and then they said, oh, all right, there's no special pathogens, or these specific ones that they're looking for, that may interfere with this test and so on. So going back to that Supernate or Supernatant, so if you have a solution right here, has mixed in with solvent, and there's a solute, these particles inside. And now, this is a solution. Yeah, the solution is all blended in perfectly inside, but then here is a suspension, is when you can see these visible. And then when you have the supernatant is once you, for example, you centrifuge it or you let it settle, then you're going to have the fluid on top. That's a supernate or supernatant. Yes, and the centrifuge is basically a device for separating particles from a solution according to their size and shape and density, viscosity of the medium and road or speed. It just spins it, and then the heavy stuff goes thrown down at the bottom. So you spin this, the heavy stuff goes further out, the more momentum, and the lighter stuff gets closer in. That's spun like that. And then this one is centrifuged, where this would be spinning around. Whoops. Yeah, whoops. This would be spinning around like that, and then the heavy stuff goes down at the bottom. So large size or heavy stuff, and then medium size, small. Then there's a solvent. It gets separated, and you'll have a supernatant of small particles and the solvent. All right, that's schematic of centrifugation. And now continuing further in the paper, so human airway epithelial cells were expanded on plastic substrate to generate passage 1 cells and were subsequently plated at a density of 2.5 times 10 to the power of 5 cells per well on permeable transwell COL or 12 millimeter diameter supports. All right, so let's make sense of all this. So plastic substrate right here refers to a plastic surface on which cells are grown. And expanding cells means growing cells outside of their natural environment. And passage 1 cells refer to cells grown and expanded in culture for the first time after obtaining from a primary cell source, such as a tissue organ. And plating refers to growing cells or cell dishes. Growing cells on dishes or multi-weld plates, which are used. Let's write multi-weld plates, which are used to produce more consistent and reproducible population of cells. So multi-welds just have holes in them or tubes, very, very tiny. Yeah. So here is a transwell coal support. Looks like this, and so you could have this would be to hold it and so on, but then there'll be these super tiny ones there. And they look like this. Continuing further, so here's some of these plastic substrates. And now here's a schematic of a transwell insert. So it's gonna look something like this. You have the upper compartment. You have the transwell insert. I believe that's these handles there, or this whole thing's insert. So this whole thing's insert. All right. So now you have this upper one. Now you have the microporous membrane. So those are the wells. So you have wells inside there. And there's, the lower compartment and so on. So, and this would be, so it looks like that. So you have a container there that can have the solvent or some liquid material or medium. And and then the inserts go inside. So and you can grow whatever you're trying to grow. And these are all cells. And you can grow them more consistently. It's very, very fascinating. And again, the number of cells right here, 2.5 times 10 to the power of 5. That's about, that'll be 3 would be 100,000. This would be, 3 would be 1,000. Then add another 2, 10. So this is about 250,000. Yeah, so absolutely dense right there. 250,000 cells. Very, very interesting. And now continuing further. Further in the paper. So human airway epithelial cell cultures were generated in an air-liquid interface for 4 to 6 weeks to form well-differentiated, polarized cultures resembling in vivo pseudostratified muscular. Let's try that again. Mucociliary epithelium. All right, so let's make sense of all this. So air-liquid interface cell culture, or ALI, involves growing cells in which the bottom portion are in contact with a liquid medium and the top exposed to air. And this is to better mimic in vivo conditions. And in vivo is Latin, for or derived from the word that means, or the words that mean within the living, and refers to tests done on the natural living organisms. In contrast, in vitro, Latin for within the glass, refers to tests done in a laboratory setting outside of normal biological conditions. And cellular differentiation is the process. Yeah, and this is dealing with this right here, well differentiated right here. I'll just bold this. Is the process of cellular differentiation is a process in which one cell changes from one type to another, and stem cells can differentiate into various cell types. And the further that cells differentiate, the more specific type of cell they become. So cells, you have stem cells becoming different cells, and then those ones become more specific types of cells. And cell polarity refers to spatial differences in shape, structure, and function within a cell. So you'll have a difference on one side than the other, and so on. And mucociliary refers to the combination of mucus and cilia in the respiratory system. And mucus is a sticky substance. Is a sticky substance that helps trap foreign particles, and cilia are hair-like structures on the surface of epithelial cells and beat in coordinated manner to move the mucus and trap particles up and down, up and out of the respiratory tract, as I showed earlier. And here, just teleported up above here. So there's a cilia right here. It moves in coordinated up and down, and it moves the it moves particles out there. And then usually the mucus traps particles, and then this moves it, and so on, out of the respiratory tract. All right, continuing further. So now here's a schematic of submerged this one is completely all liquid, and air-liquid interface this is B. Culture of primary human bronchial epithelial cells, HBEC, grown using porous culture inserts. So you have those inserts, as I showed earlier. So here is primary HBECs. These are just the human bronchial, again, inside the lungs. Epithelial cells, you can grow them like this in a liquid medium, or you can have this pseudostratified epithelium grown with this air interface set up like that. There's that pseudostratified type. So some of it, this is the liquid, and then the top is exposed to air. And there's the porous inserts to get those nutrients and so on to help grow. All right, very fascinating stuff. And here is a schematic of stem cell differentiation into various tissue types. You have a stem cell, it's undifferentiated, and then becomes, for example, a muscle cell. And this one can't become a fat cell, can't become a sex cell, and so on, because it's become differentiated. But only this cell can. So yes, if stem cell goes to here, and then or it become fat cell, bone cell, blood cell, nervous cell, and so on. Immune cell, epithelial cell, and there's the sex cells over there. And going further here's another schematic of that of the respiratory epithelium. So that's lining around this lower respiratory tract and the upper as well. So movement of mucus to the pharynx to get to this mouth, and you can spit it out. And this goes up and down with the cilia, and there's a particulate gets trapped in it, and this sticky substance is the mucus around it. All right, very interesting. As a mucus layer, cilia, and then a ciliated columnar epithelial cell, and then you have the lamina propria, or loose connective tissue, and basement membrane underneath it. This over here, underneath the cells. All right, so illustration depicting the respiratory epithelium. Very interesting. All right, so now let's continue further in the paper. So prior to infection, apical surfaces, or the top parts of them, of the surfaces of the human airway epithelial cells were washed 3 times with phosphate buffered saline, and then 150 microliters of supernatant from bronchoalveolar lavage fluid samples was inoculated onto the apical surface of the cell culture. So you have the cell cultures, they get washed 3 times with this phosphate buffered saline solutions. And then you have this 150 microliters of the supernatant from those airway fluid samples. And then you would, quote, inoculate or, quote, infect those cell cultures. All right, so again, everything just assumed it's an infection. Apical refers to the top part of the cells which extend into the open airway. And over here, a microliter or UL, this is a microliter symbol right there, is just 10 to the negative 6 liters, which is equivalent to a cubic millimeter or millimeter cubed. And you could draw it for millimeters. Millimeters about this. Let's see. Yeah, this is like more than a millimeter. See how big a millimeter would be. So this is several millimeters right here. Is a microliter like that. And here we have 150 of those. Yeah, so if you do that, it would probably be like, let's say something like this, I would assume. This amount of fluid or less. Yeah. Yeah, something like that. And 150 microliters is equal to 0.15 milliliters. Yeah, so even smaller than that maybe. Just a little tiny puddle. Like that maybe is more likely. All right, so MES note, how does the paper know there is a, quote, virus in which they are, quote, infecting the cell cultures with? So they just take from these fluid samples, they take the supernatant, and then they just place it onto these cultures. Yeah, so then the apical surface of these cultures. So they make the cultures, and then they would take the supernatant and then inoculate onto the top of the cell cultures. How do they know? That's a good question. All right, so now continuing further on the paper. So after a 2-hour incubation at 37 degrees Celsius, Unbound virus was removed by washing with 500 microliters. And note here the paper uses lowercase l, but I'm used to the uppercase L. And here I actually changed it for them here. I'll put it back here. So they use UL like that. I'm just used to writing microliter and capital L always. So yes, that's just a way I've been working with. But yeah, so they mean the same thing. So yeah, so after a 2-hour incubation at 37 degrees, Unbound virus was removed by washing with 500 microliters of phosphate buffer saline for 10 minutes. So you're washing it for 10 minutes after you grow this for 2 hours. And then human airway epithelial cells were maintained in an air-liquid interface incubated at 37 degrees with 5% carbon dioxide. And then every 48 hours, 150 microliters of phosphate buffered saline was applied to the apical surfaces of the human airway epithelial cells. And after 10 minutes of incubation at 37 degrees Celsius, the samples were harvested. And then pseudostratified mucociliary epithelium cells. Yeah, so pseudostratified mucociliary epithelium cells were maintained in this environment, and apical samples were passaged to a 1:3 diluted or 1:3 diluted vial stock to new cells. All right, let's make sense of all this now. An incubator maintains optimal temperature, humidity, and other conditions to grow and maintain cell cultures. And 37 degrees is typically chosen because it approximates the normal body temperature of humans. And serial passage, referring to this passage right here, is the process of growing bacteria or a, quote, virus in iterations by growing in one environment, then removing a portion of the population and putting it into a new environment. And this phrase, 1:3 diluted vial stock to new cells, means one part of the original sample was mixed with 3 parts of the new culture medium. Yeah, so basically they were diluting the environment. So they would dilute these apical samples, passaged or grown in another environment that is at diluted at this ratio 1:3. So 3 parts of the new culture medium versus 1 part of the original. All right, continuing further in the paper. So the cells were monitored daily with light microscopy for cytopathic effects, and with RT-PCR, or the reverse transcription PCR, for the presence of viral nucleic acid in the supernatant. After 3 passages, apical samples and human airway epithelial cells were prepared for transmission electron microscopy. All right, so they grow this 3 times. So put this. So you would grow it, then take some out, put it in a new environment, and then grow it, and then take it out, put it in, to do this 3 times total. All right. Then light microscopy or optical microscopy refers to microscopes that use visible light in a system of lenses to magnify images. And cytopathic effect or cytopathogenic effect or CPE refers to structural changes in host cells, quote, caused by viral infection, such as lysis, which is the breaking down of the cell membrane, or such as cell death without lysis due to an inability to replicate. Interesting. So basically cell death or cell breakdown and so on, cell membrane breakdown. And the assumption is it's from a virus. Interesting. All right, so now let's go over a summary of isolating the, quote, virus. So let's quickly summarize the, quote, virus isolation methodology used in this paper. So human airway fluid samples were collected from patients and mixed with a chemical preservative or fetal bovine serum, and the samples were spun really fast in a centrifuge to remove cellular debris, and the supernatant was then removed and used to, quote, infect human airway epithelial cells obtained from lung cancer surgery patients and grown as a cell culture on a plastic surface. And here's the steps in this inoculation. So prior to, quote, supernatant infection, the top part of the human airway epithelial cells, or the apical surfaces, was washed 3 times with a water-based salt solution, or PBS, phosphate buffered saline. And then 0.15 milliliters of supernatant was used in the, quote, infection, and was placed on the apical surfaces, or the top surfaces of the cell cultures. And then after 2 hours of incubation at 37 degrees, which is again normal human body temperature, with 5% CO2 in an air-liquid interface, unbound virus was removed by washing with 0.50 milliliters of PBS for 10 minutes. And this was at 500 microliters. I'm just going to call it, you convert it to 0.5 or half a milliliter. Then, PBS for 10 minutes. Interesting. Then every 48 hours, 0.15 milliliters of PBS. And I like using milliliters, just it's easier to grasp than microliters, for me at least. So 0.5 milliliters of PBS was applied to the apical surfaces of the cell cultures, then, quote, incubated or grown at 37 degrees for 10 minutes, then harvested, i.e. some apical surfaces of the cell cultures were removed from the culture. Then pseudostratified musculary or mucociliary epithelium cells were maintained in the above environment. The removed/harvested apical cells were diluted at a ratio of 1:3 to a new cell culture. And the new cell culture was monitored daily with an optical microscope for cell breakdown or cell death, aka cytopathic effects. And the supernatant of the new cell cultures was monitored with RT-PCR, reverse transcription PCR, for the presence of, quote, viral nucleic acid. And then this above procedure right here was done 2 more times, aka 3 total passages, starting from the supernatant at the last step. So you get the supernatant right here of the new cell culture, and then infecting a new set of human airway epithelial cells at the diluted setup here. Actually, no, the diluted is already done this in this step. So then they would take this and infect a new one, and then go back here, then dilute, and then monitor. And then do this 2 more times total, so that would be 3 total passages. Interesting. So last up, then quote, infecting a new set of human airway epithelial cells. All right, so after these 3 total passages, the apical cells, or the supernatant, it basically means a supernatant right here. You just take the top part, and bottom human airway epithelial cells were prepared for transmission electron microscopy, or TEM.